Tyus fish. All right. Now, this is not a Tyus fish, okay? In fact, these are beluga whales. This is a picture I took a couple weeks ago at the Georgia Aquarium. These are
been
whales.
there. Isn't that
Been,
like the
it's
biggest
awesome.
aquarium
Yeah.
or something?
Huh?
Isn't that like supposed to be the biggest aquarium in America?
Is it? I don't know.
I thought so.
I'd be surprised if Monterey Bay is not bigger, or Monterey Bay, excuse me, than California. Yeah, because that's huge.
It's big though, and it's an awesome aquarium. Anyway, so this is not a Tyus fish. That's a whale. This is a Tyus fish. Okay. So the Tyus fish has never been seen before. So we do all of the normal sort of morphological experiments, right? We write down how big the fish is, what the color of the scales are. We look at the bone structure. We cut them open and look at organs. We do all the things that we can see with our eyes. But that's old school. We have to do that because it's a new organism. But we also want to take it to the newest school using bioinformatics. So what's going to be the first step on analyzing this animal?
It's the one in Georgia, and then it's the bay.
Okay.
Yeah,
the first step in getting information about this organism is obviously getting this DNA. So what kind of cells should we get DNA from?
What do you mean?
The DNA that we're going to use to analyze this animal, where should they come from in the animal? Guess, anybody?
Fin.
Scales?
Fin.
Fin?
The blood.
Head?
Mouth.
Blood?
Mouth.
The answer is it doesn't matter, right? Because DNA is the same in every cell of the animal. Okay? They have the exact same copy of DNA in every cell. There is an exception to that. There's one kind of cell that doesn't have all of the DNA. Anybody know? Or Snickers? Anybody know?
Wait, what?
Eyes, no. What kind of cell doesn't have all of the DNA like the rest of the cells?
Reproductive cells.
What's that?
Reproductive cells.
Reproductive cells. Good. So heads up. So yeah, sperm and egg cells, right? Because they're haploid
Yeah,
and
it's just there.
humans, but we don't know what they are in the
That's
Tyus
a great
fish.
picture. That's a great picture.
Did you see the one before it? There you go.
Okay, so we collect DNA from the Tyus fish, okay? There the DNA is.
Oh, what that?
We take the DNA out of the cells. It doesn't matter what kind of cell. Blood is easy to work with, so let's go with blood. We put it into a machine that does the PCR. It takes that DNA and multiplies it by millions of, makes millions of copies. And then we put it into another machine called a sequencer, which gives us a display like this. That display, I'll zoom in a little bit. That display is what really gives us the sequence of the DNA.
You guys see those
that
letters
anytime
there?
there's a green peak, you see an A. Anytime a black peak, you see a T.
G.
So forth, okay?
A G.
So for the purposes of this, can you grab the light?
Yeah.
For the purposes of this activity, we're going to invent our own DNA.
Why can't we just like take our own DNA?
It's going to be, alright, it's going to be about...
I want to go to the one in California. Wouldn't that be cool? What is it? What aquarium is in California?
Monterey Bay.
It's the second biggest. I was right.
Okay, guys. So we're gonna make a sequence of a DNA sequence that has 25 bases. Remember that the bases are either an A, a T, a C, or a G, right? Adenine, guanine, thymine, and cytosine.
Cytosine, yeah.
So we'll start here and just give me either an A, a T, a C, or a G, and we'll go down the road. A, C, T.
G.
You said what? C. Oh.
Cat, no.
A. Speak up, I can't hear you.
C.
C.
T.
G.
C. C.
What?
C.
Who said, you said G. So you said A.
Somebody over there said G.
G.
C.
C. Please keep going.
Pay attention.
Dem.
Wait, oh, A.
This is gonna be an ugly DNA strand.
Where? G.
G.
Oh, that's just
A.
You.
Say something.
Oh, C.
There's a lot of A's and C's. Anybody gonna say a T?
Show just did. After me.
I'm saying. We're gonna skip yours.
Thanks.
G. Double Y.
See? All right, good. So everybody write that down. Either in your Twitter account, I don't care where you type it out. I would suggest, though, that you open up like a Word document or a Notepad or a WordPad document, something like that. But type it out exactly as it is, okay?
Are we gonna see what it would look like?
We're going to see what it is. Now, has anybody ever heard of a codon, also known as a triplet?
Isn't
You know
that
what a codon is?
the three?
Exactly.
Yeah.
Exactly. So if you look at the first three, that's called a codon. And then the next three will be the second codon. And the next three will be the third codon. Right? Y'all got that? So these first two rows, you're going to translate this DNA into protein, into amino acid sequence, based on the sequence just as it is. You two are going to do the same thing, but you're going to start with the second amino acid, that C, and that's going to be your first codon. Okay, so instead of ACA, you're going to have CAC, TGA, GCA, and so forth. The last row over here, you're going to start with the third letter, okay?
So what are we doing?
First two rows are doing it right from the beginning.
What, just dropping the A?
ACA, CTG, and so forth.
Some people put spaces between each three, or each triplet that they're doing, make it easier to see.
It's the same sequence, right? The sequence doesn't change. What changes is the first letter that you start with.
Wait, I have a letter that's out.
You have 25 letters, right?
So I have 3, 3, 3, 1.
Yeah, so this dropped to 1. That's fine. First thing is make sure everybody has the sequence written down or typed in, because I'm about to bring the screen back down.
What does this side do? What does this side do?
This row does, these two rows do the same thing, starting with C. So your first codon is CAC, your next codon is TGA, and so forth. What we're doing is called a frame shift. The reason why we're doing this is because when you're translating DNA into an amino acid sequence, it may not be, the DNA sequence you got out of the sequencer may not start with the first letter in the gene. So this may be the first letter, this may be the first letter, this may be the first letter of that gene. We don't know. So what you do is you do all three, and then you see which one is the best for the gene.
To be a match for something else.
That's right. Yep. To be a match for something else when we blast.
Are we supposed to like match it with its pair?
No, not yet. Does everybody have a sequence written down? If not, get it from your neighbor. It shouldn't have taken you that long just to type out that sequence.
I'm a
Just type out that sequence.
Jet's Pizza and definitely enough.
Get it from your neighbor.
My life, that is my perfect.
I need 2 more.
I don't know why, but you're right.
Okay, so we've gone in, we've gotten our DNA, ACCs and Gs. We've gone through and made a sequence. Now we're going to use this. Have anybody ever seen this chart before?
Yep, that's the secondary basis.
All right, so you know how to use it then.
Yes, I do.
All right, it's very easy, okay? What you're going to do is start with the first letter of the first codon. Remember, for this row, it's the third letter, so you're talking about an A. For you two guys, you're starting with the C, right? And for U2 rows, you're talking about the A.
A.
Alright? So find your first base over here. Okay? Let's say the first base is C. You find the row that is associated with C. See that? Now you find the second letter. Well, let's just start with the first. A is here. Okay? This is the row for A. Now you find C in the second base across the top. So boom. So we know we're working in this block somewhere, right? Y'all follow that?
Yeah.
So we got A, we got C, and then you find the third letter, which in your case is A. A is the third one down. A, C, A. Okay? So that codon is a T. So then type T. That's the first amino acid of your sequence.
Like underneath it?
That's fine, just in a new row, wherever. Okay? And then you go to the next codon. Do the same thing and get that amino acid. So by the end, you guys should have 8 amino acids. You guys follow that? You're doing the same thing.
Yeah.
So you're going to start with C.
Wait, isn't U the same thing as T?
Oh yeah, thank you. Thank you for pointing that out. Right. Remember that when you have single-stranded DNA or RNA, T is replaced with uracil, U. Okay? So anytime you see a T, just assume that we're talking about U. Okay? So you two rows are going to start with the letter C, right? You're going to find C. The next one is what? It's A. So we know we're working in this block somewhere. What's the third one?
C.
C is another C. So CAC is a histidine. It's an H. So
We're
put an
supposed
H
to just
in
put
your-
the letter or just the
Just the letter, not the whole word, just the letter. You guys understand what to do? You guys are going to start with that third letter, which is also an A, right? What's next after that? C. And then what's after that? T. So ACT or ACU is a T. It's a 3 of them.
No.
See that?
You got the sequence written down then?
Where? Does one of your friends has it?
What does stop
Sorry,
codon
cover it up
mean?
now about the print.
Good question. Stop codon, when you get to that codon, that means that's the end of a gene. Okay? Anytime you come up to a UAA or a TAA, that's the end of the gene.
Well, CTA also.
CTA, same thing. Well, not CTA, but U.
No, TGA, my bad.
Yeah, TGA and TGA. The way they taught us to remember that was you are away, you are gone, and you go away. As a way to remember that. That's
So do we
stop
not
codon.
do the...
So if you run into a stop codon, just type the word stop. That's the end of your gene. You can stop right there.
So I have TLSRL stop.
Okay, good. If you get that far, where you have now translated your DNA sequence into an amino acid sequence, the next thing I want you to do is to Google something called NCBI BLAST. Okay?
Is it together?
Yeah, you can put it together. That's it. And when you get there, click on Protein BLAST.
NCBI blast.
BLAST is the basic local alignment search tool. What BLAST does is it allows you to put your sequence, your amino acid sequence, and search it in a huge database of all known genes and sequences.
What if we just made a new one?
And that's what I'm saying. We just made a new one, but I guarantee you that it exists somewhere. So we're trying to find out what it is.
Can we make a new one though?
Yeah, you can make additional ones if you get that already. But before you do, find out what the first one was.
Where do we put it in at?
Did you see Protein BLAST?
Yeah, I'm on it.
Okay, see the top, there's a box that you cut and paste into.
Right here?
Yep. Where it says Enter Query Sequence.
TLSR? Do I have to have stop?
No, don't take stop. Once you do that, go to the bottom of the screen and hit BLAST. And that's what actually searches. That's what starts the search.
Is this it? This what I said?
Yep.
Blast!
You'll notice, Landon, one thing that you can do is you're blasting the amino acid sequence, right? You're searching the amino acid sequence. That sequence has been translated from DNA. So you should be able to blast and search the DNA sequence and get the exact same answer.
No putative conserved domain has been detected.
Yeah, that's what you want to copy and paste into the thing.
Oh, there it goes.
Okay, so go down here to protein blast. And then copy your sequence that you made, paste it into there, and then go down to the bottom and hit blast.
Speaking of blast,
There's only two. Oh, there's an extra 2 letters.
Oh, really?
No. Just
A blast in the
You're kind of
glass.
out of it.
remember, guys, you guys have just translated 24 nucleotide bases, or 25, right? Remember, people had to do this by hand, and they were dealing with genes that were, cytochrome C is 40,000 bases.
close to that, yeah.
So think about having to do this for 40,000 bases.
How do I read this?
And if you have any one base wrong, it throws off the whole sequence for the whole rest of the gene. You see that?
Yeah, don't worry about that.
So that's the power of bioinformatics. Now there are applications, there are apps that will do this for you. Okay, you can give it a DNA sequence that's 1 million bases long, and in a matter of seconds, it'll give you the DNA. The translated amino acid sequence.
What does it mean? I don't know what this means.
I know. No, I agree. That's why I'm laughing, because I look at it, I'm like, what?
Scroll down. Yep. There you go. DNA repair helicase. So that's a protein. So what you have right there is a protein that allows cells to repair their own DNA. That's the function of the protein that we created. And if you translated it right, everybody else in these 2 rows should have that same.
Go right here where it says protein blast.
The second one, right?
What'd you get?
The second one.
See that?
I mean, that looks right, right?
ACA,
NCB.
threonine,
You just copy and paste.
C2G, is leucine, AGC, is serine,
Do a search for
AGA,
I'm sorry, NCBI.
arginine,
Blast. Or just,
Yeah.
How you
See
get
that little
to
spot?
it?
I think you enter the code. I don't know, because I haven't got there
Zia, how you
choline,
get to it?
Well, CTA is
Oh, go back.
C, right, leucine.
Yeah, over here.
Under the name, it's like a small name.
Over here we got
Click on it.
Quantumate, quantumate dehydrogenase.
What are we doing to get there?
What is it?
Where's that?
So,
Yeah,
now,
Do we put in that 25 color in here?
I didn't hear you.
Do we put in that 25 letter
You
color?
can do that. Now keep in mind, that's great. Keep in mind that what you're on is the protein blast. It is what you use to search all the protein databases. If you want to search the DNA databases, you go to what they call nucleotide blast. So if you go back one, in fact, right here at the top of the page, this blast N. Blast N is blast nucleotides. We're under blast P, which is blast protein. Or you can blast nucleotides. You can type a nucleotide sequence and search that against proteins, and that's blast X. There's a bunch of other stuff that you can do. Translated proteins and all sorts of stuff.
Why? Because I've wanted to know how to use this site for forever, and now I understand a little bit.
All the genes that it comes back, this long list of genes, all the genes that it searches are all in a place called the gene bank. It's actually spelled GenBank, G-E-N bank. It's a database, a bank of all known genes everywhere. So in our laboratory, for instance, we didn't create, but we discovered a new gene in the brain of a bird. So, and in fact, the brain of a guinea fowl. And that's what I won that $200 prize for, because I went and presented that work in Texas. It's a gene that we know is made in the small intestine, and people always thought that it's probably made in the brain too, in the hypothalamus, but nobody's been able to prove it. One lab was able to prove it in a mouse, but nobody else has been able to prove it in any other animal. So we were able to prove it in in a bird. And how do we know what that gene is? Because I went in, I got DNA, and I blasted it, and it comes back as, it's GIP is the name of the gene, okay?
That sounds like a rapper's name.
GIP.
My name is GF.
Notorious GIP.
No.
So the other thing that this will allow us to do is to get some idea on how closely related our gene in the Tyus fish is to another organism. So Landon, where you have that the DNA repair helicase, I want you to scroll down to the description, and it should tell you which organism it's related to and how closely related it is.
What, this?
So in fact, give me the which computer are we on, Taylor?
Hey,
Yeah.
Jello, my.
You can move it. You can shift
Capsulus.
if you need to.
What'd you type in?
Give me the
The sequence?
yeah, give me the sequence that you came up with.